DNA replication fork. DNA is made of nucleotides that carry genetic information and is arranged in two complementary strands forming a double helix. Nucleotides are formed by a phosphate group, a sugar, and a nitrogenous base that is bonded to its complementary base through hydrogen bonding. DNA replication is the process of producing two identical copies of DNA, in which each strand acts as a template for the synthesis of a new complementary daughter strand. The first step of DNA replication is separating the two strands. Gyrase is an enzyme that helps unwind DNA by relaxing the supercoiled double helix structure. Separating the two strands is a process that occurs through the help of an enzyme known as DNA helicase, which is responsible for breaking the hydrogen bonds that occur between the nitrogenous bases, resulting in the formation of a replication fork. The replication fork is the region of DNA synthesis where the parental strands separate and two new daughter strands elongate. These separated strands act as a template for creating a new strand of DNA. As it was previously mentioned, the formation of the replication fork results in two new DNA strands that run in opposite direction. This direction indicates how replication will occur through assigning the leading and the lagging strands. The leading strands runs in a 5' to 3' direction, while the lagging strands runs in a 3' to 5' direction. It's important to note that DNA replication occurs in a 5' to 3' direction. To continue replication after the formation of the two new strands, an enzyme known as RNA primase starts the replication process by creating small pieces of RNA called a primer that indicates the starting point of the construction of the new DNA, or its origin of replication. Next, DNA polymerase III attaches to the RNA primer and attaches new complementary bases in the 5' to 3' direction, making the replication along the leading strand continuous. Because the lagging strand runs in the 3' to 5' direction, replication is not continuous. Therefore, to replicate the strand, DNA polymerase III makes the new strand in a series of small chunks known as Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase III then adds a short row of DNA bases in the 5' to 3' direction. This process is repeated until there are numerous fragments along the strand. After there are numerous fragments, an enzyme known as DNA polymerase I removes the RNA primers on both strands and replaces them with DNA nucleotides. Finally, ligase seals up the fragments together through creating a phosphodiester bond between the 3' OH end and the 5' phosphate group, thus creating two semi-conservative strands.